everyone. Welcome to this new episode of our Student Spotlight series, where we're able to showcase some of the new talent that our past students have been exhibiting. And I'm Daphne. I'm one of the podcast hosts here at CCIR, and it's our pleasure today to have you with us. So why don't you tell us a little bit about yourself, like how old you are, your name, how long you've been with CCIR, what grade you're in, like these kinds of things.
All right, so my name is Arshmeet. I'm 16 years old, turning 17 in December. I was with CCIR for the summer term of this year, so that's my experience here.
Which grade are you in?
Oh, just graduated, and I'm actually in my first year of college now. Yeah.
You're only 16 in your first year of college.
Yeah, I had sort of like a untraditional education path. So I was dual enrolled throughout high school, at a community college, which for all the people in the UK is like a thing you can do in America. And then now I'm doing like my first year of college.
That's amazing. Which college are you going to?
It's just community college.
Oh,
So,
wow. And where are you from? Where do you live?
I'm from the Bay Area,
In
specifically from
California?
San Jose. Yeah, in
That's very,
California.
I'm from New York City, so that's very cool. The two coasts kind of, that's brilliant. That's very impressive that you're in your first year of college already,
Thank you.
only at 16. Wow. Um, Amazing. So, yeah, I'm just curious to hear a little bit about your project. Which professor were you paired with for the program?
I was paired with Professor Zarkin, and I was in the computational genetics and drug discovery program.
Wow, that's really, really cool. Yeah, I would love to hear a bit about your research paper. So what was your topic? What did you focus on?
So my topic was focusing on antimicrobial resistance, specifically on vancomycin resistance in two species of bacteria. So Staphylococcus aureus and Enterococci. So that's what I was sort of looking at as like the broad topic. Yeah. And specifically, I was looking at these two studies that had been done. One was on a patient in New York in 2004 who had contracted staph and enterococci infection at the same time. And there had been previous studies looking at like what the similarities and differences were in the vanA operon within that one patient. And then I also found data from another patient who had received a liver transmission some years later from someone who had an enterococci infection, a vancomycin-resistant enterococci infection. And the point of that study or collecting that data was the researchers were trying to figure out like whether the transmission of the bacteria had been from the liver transplant itself or how the hospital had been sort of handling the liver transplant. So if it was hospital transmitted or like transplant transmitted. So I took data from those two studies and I sort of wanted to compare the vanA genes between all the different strains of bacteria in both patients. And I should probably give some background onto like what the vanA gene is. The vanA gene is a gene within the vanA operon in bacteria that carry the vanA operon, which would make you like resistant to a drug called vancomycin. And its main role is just to produce, basically, in the most simple sense, like a type of protein that would make it harder for the antibiotic to bind. Yep.
Wow, that is really fascinating. That's amazing. How did you decide to choose this research topic in particular?
So I was looking at basically in the first few weeks of the course, we were reading review papers about antimicrobial resistance, specifically in staph. And I found this really interesting because there had only been, I think, 12 cases in the entire United States of vancomycin resistant infections of staph. Um, so I was just really interested in like how much research had been done on this. And also, vancomycin is considered to be like a last resort sort of antibiotic for these sorts of infections that are very like resistant to things like other drugs like methicillin. So I was interested to see, like, understand more about like how that works, like what the mechanism is, and then also in the bacteria that have developed resistance, like what the genetic diversity looks like.
Wow, that's really cool. Um, yeah, and so kind of walk me through how your research paper ended up being what it is today. Like, walk me through your whole kind of process from the idea coming into your head to, eventually research having been done, all the pieces coming together, you actually writing it. How did you formulate the plan under which to construct your research, and what did those steps kind of look like?
Yeah, so for the first couple of weeks of the course, we didn't really do any research. We were just sort of learning about the material, which I feel like was pretty helpful to sort of get to choose our topic really freely. We spend a lot of time in TA sessions in the first couple, just sort of reading research papers, and that's where I stumbled across the idea. And then after that, the first step that we did was actually writing the introduction. So this included like getting some background on whatever research had been done before in this area, and then sort of like understanding how the drug works, vancomycin, against bacteria, and like shortening our aims section. So defining like aims for the paper, like what do we want to address in this paper? So that was probably one of the harder parts of doing the research, was trying to figure out like what exactly I wanted the paper to address and how I could do that with the types of analyses that we were learning in the course. So once I had that down, sort of the aims of my paper, um, I decided on trying to find out the degree of conservation of vanA across all the strains that I had, like all the data that I would collect. So the degree of conservation would just mean like how similar the genes are between all the different strains. And if they're like very similar, it's likely that they're like highly conserved. So that's what I like my final aim was. And then after that, the first step was to do data collection, um, which was it was a little hard because there have only been like 12 cases of these infections in the US. And I think I found a pretty decent amount of bacterial strains. I got in total 27. So I collected those from NCBI, downloaded them, and then sort of like started doing some quality testing on the genomes. So I uploaded them onto a site called Pathogen Watch, which basically tells you if you have contamination. So what that means is you upload the genome, it labels which species the genome is, and then you should match that to like whatever is written on NCBI as like confirmation that you have the right species.
Hmm.
I also ran like cost analysis and then like just some sort of things to make sure my genome quality was good.
Amazing. And for those of us who don't know, can you define cost analysis for us?
Yeah, so Quast is this like command. It's I don't know exactly what to call it. It's like a program. You it's like a command line tool. You download it and then you upload your FOSTA files of your genomes. Um, and it will basically give you a set of metrics. So these could be like the total genome length, and then you could sort of match that to the average genome length for that specific species. So for example, um, if I put in like one of my strains and it gave me a genome length of like 2.8, um, I forgot actually how much specifically it is, but like it'll give me a genome size, how long that genome is, and then I can compare it to like how long it's supposed to be, like the average for that species. So that's one metric you get. You can get like other metrics like N50, or like the size of contigs. So contigs are these basically like pieces, I guess, is how I came to understand them, that the genome is like split into when sequencing or like when being put into the bioinformatics tools. So you can get the number of contigs, you can get a bunch of metrics that are really helpful to understand like how good the quality of your genomes that you downloaded is.
Hmm. Very interesting. Okay, I understand. I guess my next question for you is how did you, well, you kind of walked us through how you picked your topic. I'm also wondering how you read up on other scientists conducting similar research and how you set yourself against the already existing body of work. Because I know staph is quite a popular bacteria to conduct research about. So I'm wondering how, like, what you read about to prepare yourself for this research project and why your project kind of is set apart from the rest of the existing scholarship already out in the field. I'm curious on your thoughts on this.
Right. So, as I already mentioned, like I chose sort of a niche topic, sort of something that didn't have too much research on it. Obviously, there's a lot of research on like vancomycin resistant infections, but staph specifically, only like 12 cases. So maximum you're gonna have like maybe 12 to 24 studies that you have to read on that, right? Um, so I sort of went in like going for a niche because I really wanted to conduct original research. Like to me, a lot of the fun of science, like getting to find something new or like something adding to a field, basically. So I went on NCBI and where I downloaded the genomes, I basically like looked at those papers. They usually have the paper that the genomes are provided from. And I read those, and then I read some review papers, just like meta-analyses on vancomycin resistance in general. And through reading those and reading the more specific papers, that's how I was able to find like a research gap of like what have previous studies addressed, and then what could my study reasonably sort of feasibly address. And um, that's sort of how I arrived at like my aims as well, because I saw that like the specific thing that I was looking at, conservation of vanA, it had been studied before, but like in other papers that I was looking at, it was studied before in like an animal infection or studied before in like different cases. So I sort of saw that as like, oh, this can be a potential research gap that I can fill. Yeah.
That's really interesting. Yeah. Yeah, because I know not a lot of us know about vancomycin and how few cases there are, like, kind of in your chosen area of study. And that must have been really challenging to only work with that small a sample size. Yeah, that's really interesting. Do you hope to get your paper published one day soon?
Yeah, I'm I'm hoping to get it published. I'm working on just putting like the finishing sort of touches on it, and then I want to try and start sending it to journals. Yeah.
Amazing. Yeah, I guess my next question for you is kind of like if you had any tips and tricks for anyone starting a similar research paper, um, maybe at CCIR or just in general, what are some kind of words of wisdom that you'd give to your peers or those coming up just behind you?
I'd say get started pretty early on. Like, I feel like the like finding out what you want to do or like sort of finding the research gap that you want to fill, it actually takes a lot more time than doing like the analysis itself. So I would like as soon as you get into the course, I would start reading like review papers that you're interested in and sort of looking around the topic, looking around the area of like what do you want to do and what data can you find and what kind of project can be feasible for you. Um, so I would start that pretty early because I feel like that's the part of the project that takes the most time. So that's the number one tip that I have. Yeah.
That's really great. Yeah, thank you. I'm also wondering, I know you were saying that you've already kind of finished high school, which is incredible, and you're doing community college right now and taking courses, like actual college level courses, which is amazing for your 16-year-old age. I'm just so impressed by this. Um, but my other question is kind of, had you done one-on-one work with a professor in a similar way before, or was that quite a new academic structure in comparison to maybe larger group sizes in high school, more curriculum based exercises and projects and reading assignments and less freedom in the sense of conducting your own research, crafting your own paper, and your own step-by-step guide to your own material. Was that and a different academic structure? And if so, how was that for you? Was it challenging? Was it interesting? Was it exciting? I'm kind of curious to hear about this side of it.
Yeah, so it's really interesting actually. I'd say like the academic structure and the way that things were structured, I feel like there was always opportunity for us to like struggle a little bit, but then get some help, you know? Like there was always like we could book a TA session, and then of course every week we would also have a TA session. So I felt very like supported in that way. I'd say comparing it to like other academic experiences, I was also like homeschooled throughout like high school. So basically I basically did high school at a community college. Um just taking like courses at a slower unit pace basically. And then I got my like California high school exit exam to like officially be out of high school and then start doing college courses as a college student. So I would say like that's the only thing that I can really compare my experience to. So I can't really compare it to like a traditional high school setting, but I would say the structure was actually like quite similar. I've had projects in classes before where you start at the start of the semester, and by the end of the semester, you have to deliver like a paper or some sort of like project that you've built. And I really like the format of like every 2 weeks or every week, we would just have like a meeting to see like where I'm at, what I've completed, and then like what I sort of need help with. Um, yeah, I'd say like it's you definitely need to have like the skill to like struggle with things a lot and
Mhm.
then sort of figure stuff out on your own. But I feel like most people have that skill in high school anyway. So I guess my answer to that question is like the academic setting isn't very like intimidating or anything. It's just a
Mm.
little bit different, I guess, if you're coming from high school.
Yeah, definitely. I feel like this side of pursuing your own research and
Mm.
forging your path almost in terms of what you envision your paper eventually looking like. It's quite like a special thing, kind of like how you're saying. So, no, definitely. Um, I'm also curious if you could choose to do another research project like right at this very moment, and it cannot be the one you've just done, what other topics are you interested in? Like, how would you structure a new research project potentially?
If I'm gonna do a new research project, I would really like to like do something that requires like lab experience actually. I've been looking into like applying for internships and like lab opportunities and stuff in my area. So something that I'm really interested in right now is like CRISPR, and there's a couple of laboratories around me that are working on stuff involving CRISPR. I'm also really interested in like all sorts of bioengineering things, but mostly in the fields of like genetics. Um, and there's a lot of like different genetics programs that I'm looking at in my area for like summer research next year. And yeah, so I'd probably try to get like more hands-on experience because I really liked the computational research, but I feel like now that I've done computational research, I kind of want to like experience the more like
Mm.
hands-on thing. Yeah.
So kind of stepping away from the laptop and going more into the lab almost, like more
Yeah.
kind of physical, I guess. No, that's
Mhm.
really interesting. That's brilliant. Yeah, I think that kind of brings us to the end of our interview. Thank you so much for having the chance to talk with me today.
Thank you so much for inviting me.